Welcome to Farming Matters, the storytelling series where you get to hear from innovative farmers and ranchers across the North Central region. Today, we're going to hear from Alan Stovall, a beekeeper who's trying some innovative ways to improve beehive health based on what the bees are foraging on. Every one of our presenters is someone who's received a SARE grant, and SARE stands for Sustainable Agriculture Research Education. And basically, we give grants to farmers and ranchers who want to try out innovative solutions to problems that folks face all around our region and then share what they learn. And this is just one way that they share. Here at SARE, our goal is to advance innovation to the whole of American agriculture, sustainable innovations for everyone. And so without further ado, let's hear from Alan.
Hi, my name is Alan Stovall. I am a beekeeper here in Salina, Kansas. Um, we applied for our SARE grant a couple years ago, and it was a 2-year grant that we were working with. So what the grant was based towards is we wanted to find out if a certain type of flower would provide honey and nectar sources, pollen sources for our honeybees during the fall months of the year. We're looking at like July, September, October, and to help us with the dearth during those months to be able to provide a sustainable food source for our honeybees with those flowers. The main flower that we were working with is called Silphium, or it's also known as Silflower. The sites that we did have our bees on that had that Silflower did quite well with utilizing that plant. So the grant was for 40 hives at different locations. So we placed 10 hives at 4 different locations. 20 of the hives had no access or no visible or notable access to the Silphium flower that we were doing our research with, because we needed to have a what do you call that? A test,
Like a control.
a control group. Yes, thank you very much. So we were using those 20 hives as a control group versus the other 20 hives that we had placed on the Silphium flowers. 2 of the sites had those Silphium flowers within 20 to 30 feet of our beehives, and there was at least 2 to 3 acres of that flower planted to be able to give them access to see how well they were doing with it. The first year that we started out with all this was just a building year. With most new hives, the bees will need to build out all the comb and get all that taken care of. And that was what we really focused on with those 40 colonies that first year, was getting those hives built out. So the second year, we could get a real accurate kind of idea of what one field versus another field is doing for us with a complete solid 40 hives. And all 40 hives were very strong in the second year. Whether I had to replace bees or not, some of them didn't make it through the winter, which we really struggle with that here in Kansas, is getting our bees through the winter. But if they didn't make it through the winter, once the hives are drawn out, it's very easy to add a colony to that hive, or that old hive, or the drawn out hive, and they will take that and work with that very quickly. So the other plant that we were working with is Sainfoin, which is a type of clover. Now, Sainfoin comes in the spring, and honeybees are really, they love that Sainfoin. Kind of like, I would say, like any other clovers, they'll go for the Sainfoin before they'll go for some of the other flowers in the area. It produces a really flavorful honey, and it's one of the first flowers that your bees are going to be able to get to actually start pulling nectar from at the beginning of the spring season. There's, you may see in online that don't kill the dandelions and all that type of stuff, because those are first foods. That Sainfoin is also a first food. It will actually flower multiple times throughout the spring and summer season. So it's not just kind of a one and done situation for the bees. The bees can go back to it continuously throughout that season until I'm going to say probably late July.
And did you have, so I'm trying to remember, so you had 4 sites, two were controls, and two, did they have both Sainfoin and Silflower or one of each? I forget.
Yes, both of those, the other sites, both that had the Silflower, both had the Sainfoin as well.
Okay, nice. So you were adding forage for the bees in the early season and during that dearth.
Yes,
Yeah,
yes.
neat.
So, and like I say, the Silphium, it did show some significance in that dearth period. They were pulling that Silflower and that nectar and the pollens from the Silflower quite a bit from what I'm seeing in our DNA testing. So on the left here, we have a pie chart. Now these are the fields that we had with the Silflower and the Sainfoin. As you can see, we had a significant increase in the Silflower production and the Sainfoin production that the honeybees were getting, versus the right-hand side where there was no access or no known visible access to those flowers. They didn't pull near as much from either one of those flowers. The Asaria area put about 3.5% silphium into the honey that we had tested versus the 0.6% from the rock quarry. And the rock quarry is a great big area where it's an old rock quarry and it's like turned into a pond now. So, and they have all sorts of vegetation, flowers, alfalfa, soybeans out in that area, which is great for, as the pie charts show, it's great for the honeybees, but we really were trying to focus on making sure we were finding something through that fall time, late July, August, September, October time, and hoping that the silphium would take care of that for us. Okay, so the two test sites, I would say the control sites that did not have access to the Sainfoin was the Rock Quarry and the Finch Farm. The two sites that had the Sainfoin and the silphium are going to be Charlie's and Farm. The farm is TLI's purchased land. Charlie's is an area that they are leasing from, that they're doing more research out there. So of course, if you look at what we have here in this chart, it's going to show you that in 22, we had hives out at the rock quarry, or not the rock quarry, but we had hives out at Charlie's. That's going to be the Asaria site. And we also had hives out at the Finch Farm, which was one of the private farmers that we work with. We did not have any colonies out at the main farm. We work with an organization called the Land Institute here in Salina, Kansas, and they have provided us with the Asaria, and they have a farm here. It's just a couple of miles outside of town here that they do a bunch of research with perennial plants and other types of things like that. We did not have access to that until 2023. The Rock Quarry was another private farmer that gave us access in 2023 because they had received is it NC? No, it's a grant where you can get wildflower and milkweed seeds to spread out.
Yeah, it might be
For
through NRCS.
Yeah, I think it might have been. I don't remember exactly. I have to look it back up again. But the gentleman who owned that property got that grant and asked us to place bees out there for him. Now, as far as the DNA hits, so we send off samples to Colorado each year for our honey because we want to know what they're pulling out of the local area. We want to know what flowers they're harvesting from. So what we do is we send that off, and they start analyzing the pollen in the honey. And those DNA hits are actually pollen counts that they're doing for that analysis. So that's what that means, is the DNA hits are pollen counts that when we send our honey off to Colorado, as they go through it, they count out all the pollen counts, and then they come back with they come back at us with those numbers and let us know exactly how much of the honey was soybean, how much of the honey was alfalfa versus clovers versus other asters, all that type of stuff. So that's how we were able to measure how much Sainfoin or Silphium we were getting from Charlie's in the farm versus the rock quarry and finches. Even with the grant being having concluded, we are still going to each year send off DNA samples because we want to be able to keep those records on hand and know exactly what our bees are doing with that. And not necessarily just for our own information, but if anybody else asked for that type of information, we would be able to readily provide that for them. So one of the largest things I really one of the things that stands out most for me is that those fields that had the Sainfoin and they had the Silphium, those fields produced much more honey in general than the fields that didn't have access to those, which was pretty significant. So it kind of makes me wonder what it is that those two plants are providing that's able to promote the honeybees into building more honey or gathering more nectar and more pollen for those colonies versus the other colonies, which is maybe another research study down the road.
So, Alan, are you thinking then that the fact that they had access to the nectar and pollen right away at the start of the season, and then during that dearth period, that's where that bump in honey production might have come from?
You know, it might have, but bees are so dynamic.
Hmm.
What we could be working with is the fact that they had access to the Sainfoin in the springtime may have allowed them to collect pollens and nectars that allowed that colony to super build itself,
Hmm.
of which grows your population, you get more production out of more bees. And then of course, when we hit the Silflower, that might have just even kind of boosted that a little bit more because your asters, they're going to provide quite a bit of pollen.
Yeah.
So that's kind of where I was leading with that, was thinking that maybe it helped out the colony in population,
Hmm.
which will of course help out your production when you go to start finding out how much honey you were able to get out of each colony.
Yeah. Yeah,
Um,
absolutely.
I found out personally just kind of by keeping an eye on things. I was pulling roughly a 5 gallon bucket out of the highs at the locations that had the Silflower and Sainfoin. Um, if I had two honey super boxes on it, I could get a 5 gallon bucket of honey out of each one of those hives, which is pretty good. So I was very happy with those. On the other hand, the other hives that were part of our control group, at the rock quarry, I noticed that I didn't get a whole lot of honey production out of that spot, but bee population, the hives were just really, really loaded with bees. And so that kind of makes me think that maybe they had a lot more access to pollen and not as much access to nectar. Because, of course, it's your nectar that's making your honey. So they take that nectar and they put that into the cells. When they pull nectar from the flower, it has a very high water content to it. And they dry that down to below 18% water content. And that turns that nectar is then called honey. They put capping over, and that's what seals it and makes it good. If they don't have a whole lot of nectar in their or close to them, then they may not be able to produce as much honey versus if they have more pollen. They all made it through the winter at that location very well. They even made it through this last winter very well, which was I'm very pleased with that location as far as hive survival. I'm not real sure what I need to do as far as being able to get them to start producing more honey for me. I really think a lot of all that has to do with location. My experience is I've been in several areas, and people will move their bees to areas that are pollinating while they're pollinating. We were just recently in North Carolina and talked to some beekeepers out there, and they moved their bees for 2 weeks onto sourwood trees and let them pollinate those sourwood trees. And that's all the longer those sourwood trees pollinate for. And then they take them back and put them somewhere else. One of the reasons a beekeeper would do that is because they want to be able to single source name their honey. And it is my understanding that if your honey is above 50% one pollen type, you can call it that type of honey. So if a clover honey is over 50% clover in its DNA hits, then you can call that clover honey. But if it's under that, then you can't, it's just raw wild honey.
I remember in your report, Ellen, you guys did talk about the marketing related to the either the Sainfoin or the Silflower. I don't remember which one. That one of them, just observationally, customers were asking for it by name, and that it seemed like it didn't crystallize as quickly.
Yes, the Silflower. Now, your Silflower is going to crystallize, though it's a sunflower, or it's from the sunflower family. Um, for some reason, that honey really sunflower honey can crystallize. It doesn't crystallize as fast as some other honeys. Like up in North Dakota, you get a lot of canola. That canola will crystallize like within days and turn rock solid.
Huh, I didn't know.
So Silflower doesn't do quite that quickly, or sunflowers or the aster family doesn't work quite that quickly, but it will crystallize on you. Sainfoin, like clovers, it will stay liquid much longer. But in the end, all honey crystallizes.
We just need to
So
drink more heat.
it's just kind of how it nature makes it preserve itself, I guess. I think there's one more slide here, the conclusions. So we did observe our honeybees feeding on Silflower, and Silflower doesn't appear to be the biggest percentage of their diet. Of course, there is a lot of alfalfa and a lot of soybean in our area. The bees also fed on the Sainfoin, and that wasn't a large percentage of their nectar source. Hives that had more Sainfoin pollen in their honey tended to have higher honey yields. So those colonies that had access to that Sainfoin produced more honey for us. Bees also had higher honey yields while foraging on soybeans and clover as well. And that might have been just more accessibility to those plants.
I'm thinking about the Sainfoin and the Silflower as potential crops. Why would a row crop farmer or a livestock farmer plant those? Can you speak to that at all?
So on the I don't have a lot of information with the Sainfoin, but the Silflower is actually one of the reasons it went to the Land Institute for study is because there are two known uses for Silphium. One is to use as a feed to mix into like your hay, your straw, whatever you're feeding your livestock. You can use that Silphium in there with that, and it helps them. You can actually put cattle out on it, let them graze it. Just don't let them chew all the way down to the ground, or it might kill the plant.
Sure.
The other use that you can get out of the Silphium flower is it produces a oil-bearing seed. So the seed, if you process it to pull the oils out of it, that's actually consumable for us to be able to use as like a cooking oil, like a sunflower seed oil almost. But it's Sainfoin, not the typical type of sunflower oil that you would get nowadays in your grocery store. It'd just be a different type of source from that, I guess. But the third reason, and this is why we initially started to think about this project, is because we wanted to know if that Silphium flower was going to have a third use as far as honey production for us, and to be able to help increase our honey production. By what our charts show, yes, it has helped to increase that production on those fields that they had access to that. So we were very pleased to find that out. Um, I think I'd like to see consistency through it though. And something like that, I feel would take several years to be able to make sure that we're seeing consistent numbers from that type of information.
So is that the plan? To see if you continue to plant it and see if it has that consistency?
So what we have at this point is we have currently signed another 2-year contract with the Land Institute, and we will be keeping bees on those fields for the next 2 years to be able to start tracking that information as well. And then, of course, out of the other locations, I've got 6 locations now total that I keep bees on, and ever expanding. So we do have control groups that if we wanted to try to do this again with more data, we would be able to do that with our control groups. And I wouldn't have to do any building years. That first year was we had to get ourselves built out to 40 hives, and they all had to be viable. So that was the main focus for the first year. The second year, those hives were built. They were all viable. They were all doing what they were supposed to do. At this point, I have enough hives where I don't have to worry about that first building year anymore, and we can get right into crunching numbers.
Alan, thinking about other beekeepers out there who might want to be trying something innovative like this, any words of wisdom if they want to apply for a SARE grant?
Well, the SARE grant was actually our first grant. So we applied for it the first year, and we didn't get it, which we didn't look at that as a disappointment. We took that as a learning opportunity. So we looked at our grant. We took the feedback from everybody that looked over our grant, and we refined it. And we did it. We reapplied in the second year. And in the second year, we were able to get our grant, rewrite it, refine it, make it better, resubmit it until you do get what you're going for.
Oh, that's beautiful. That's really good to hear. We have reviewers, other farmers and ranchers from all over our 12 states who review all the applications that come in, and they spend a lot of time writing out their thoughts, and it's always constructive criticism. And then, yeah, we share that back with everyone who applies. But we always worry, will people see it for what it's supposed to be, which is helping you push your idea forward. So it's great to
Absolutely.
hear that worked.
Yes. Yes. And that's exactly how we saw it. And we look at a lot of things like that. I mean, we run our own business here in Salina, Kansas with AJ Honey Farms. And I started beekeeping about 8 years ago. And when I first started, it became an absolute obsession of mine. And I, at this point, take all that information that I can be given, and I try to make our business and what we're doing here grow and be better from it. Even if, that's what it is. It's constructive criticism. It's designed to help you. It's not designed to put you down or make you feel bad about something. Evolve, learn from it, and grow.
You've answered all of my questions. It was really neat to hear about straight from you, Alan, how this all went. Thank you.
Well, thank you for having me. I appreciate the opportunity to do this.